Okay, so good afternoon. I trust that the good weather will bring more folks, but I'm sure you're all dedicated to the topic today, so we're delighted that you're here. I would remind you later when we have questions or discussions, just wait for a minute till you get the microphone, because each week there are several hundred people at NIH who are online, and they can't hear the questions, so that's why we do it that way. Okay? So I won't go through the Brooklyn Bridge. We're a little bit late, and I think you've all heard the reason why. This is the logo to cross science and medicine. And the topic of today is the next one, if I can figure out which one to push. Okay. So although single cells have functions which are delineated in specific parts or directions of those cells, and probably every cell in the body, I guess with the exception of the red blood cell, has that kind of regionalized activity, which would be defined, at least to biologists, as polarity. Physicists have a different definition of polarity, having to do with pluses and minuses. But it's probably in the Mesozoic time when single cells got together to form groups of cells, which then became organs and then became organisms and species. And that process of cells coming together to form an organ required junctions, and it required separation of one domain of the plasma membrane from the other. And that is tied up with specific functions of a cell. And so today we're going to really have a discussion of two different systems, the nervous system and the hepatocyte system, which illustrate different aspects of this overall concept of the vital importance of polarity to maintain functional integrity and life. So our first speaker today, Juan Bonifacino, received his Ph.D. in biochemistry at the University of Buenos Aires, where he still is an honorary professor. And following a postdoc fellowship with Rick Klausner, he became a permanent person here at the NIH and has been here since 1981. He's the Associate Scientific Director of the Cell Biology and Neurobiology branch in NICHD. And Juan's work is known worldwide. He is one of clearly the leading thinkers and scientists in cell biology in general, but specifically with respect to intracellular trafficking and its regulation, which is a key element in the polarization process, as you will see if you don't know already. And in recent years has focused on studies in the central nervous system, which he's going to speak about today. Now, Juan goes way beyond the basic biology, which he hopes to delineate, and then others follow along, because he reaches out and collaborates with people who come forth with diseases, not the patients, but the physicians, diseases in the beginning that are inheritable and then become focused into the pathway that he has defined. And then it gets to be broader when you begin to think of acquired diseases, drugs, viruses, and so forth, which are not so specific with respect to an individual gene being affected or its transcription or anything like that. And that's the same theme that's going to involve later with the liver. So Juan has trained a huge number, 70 or 80 investigators here at NIH who have populated the world, and we're really very grateful to you for taking the time to present.
Well, thank you very much, Win, for your very, very kind introduction and for inviting me again to speak at this course. think I spoke about 2 or 3 years ago about a different topic. And thank you all for being here this afternoon. So in the first part of this talk, I'm going to talk about mechanisms of polarized sorting in neurons, and then Wynn will talk about the liver, I guess, or epithelial cells. And so Wynn already very nicely introduced the topic of cell polarity, and he actually had an analogy of cell polarity. I have my own, which is it's the Earth. The Earth is a polarized object. It has a North Pole, and it has a South Pole. And both poles are very different. The North Pole has a sea, the Arctic Sea, and the South Pole has a continent, Antarctica, so they are very different. And there are different inhabitants in the North Pole and the South Pole. In the North Pole, there are polar bears now jumping from a chunk of ice onto the next one. And whereas in the South Pole, or close to the South Pole anyway, there are penguins. So both poles are very different, and they have different properties. So likewise, as Wynn mentioned, in our body and in nature in general, all cells have a polarized organization. They have, in many cases, a North Pole and a South Pole. And two of the most commonly studied cell types that are polarized are epithelial cells and neurons. And so epithelial cells have an apical pole, an apical domain, and they have a basal domain. And they also have lateral domains, and for many purposes, the basal and the lateral domains are considered as a single basolateral domain. So this is called apicobasal polarity. And neurons also have a similar polarized organization. They have axons and they have dendrites. In epithelial cells and in other cell types, particularly during development, there is an additional axis of polarity, which is anterior-posterior polarity, also called planar cell polarity, in which the lateral domains are different. There is an anterior domain and a posterior domain that also have different properties. And so in neurons, although this axis is not so clearly delineated, there is a role for components of the anterior-posterior polarity machine. Now, so as I said before, these domains are different. These poles are different. They have different protein and lipid compositions. They have different organelle compositions in the cytoplasm, and they have different properties, different functions in both epithelial cells and in neurons. Now, how is this polarity established and then maintained? Well, this is a very complex process, and there are many determinants of cell polarity. First of all, there are extracellular signals that tell the cells to become polarized and how they should become polarized. Signals coming from the extracellular matrix, from other cells, or from gradients of soluble mediators. For example, nerve growth factor, BDNF, Wnt, and many others. So there are extracellular signals that tell the cells how to become polarized. Those signals are received by surface receptors on the polarized cells and adhesion molecules for the extracellular matrix, for other cells, and for the soluble mediators. In addition, there are polarity modules or molecules that are intracellular and that mark the sites where one of the poles of the cell will be established. That includes proteins such as PAR, CRAMS, Scribble, Rho for apico-basal polarity in epithelial cells, and Frizzled, Van Gogh, and many others for planar cell polarity. Then there are signaling pathways that coordinate all of these processes. There is also a trafficking machinery, trafficking pathways that deliver the components to the different domains. So in the case of epithelial cells, to the apical domain or to the basolateral domain. And finally, the cytoskeleton also contributes to the establishment and maintenance of cell polarity by supporting the transport of those components to the different domains or the organization of the different domains. And in particular, the microtubule cytoskeleton and the actin cytoskeleton. Now, mutations in some of these 4 determinants have already been found to underlie various diseases, including some very common diseases such as cancer. In fact, one of the hallmarks of oncogenic transformation is the loss of the polarized organization of cells, for instance, intestinal epithelial cells. And in many cases, that's linked to mutations or to changes in the expression levels of various polarity determinants such as PTEN, PI3 kinase, scribble, and many others. But in addition, there are mutations in some of these polarity determinants in other hereditary diseases, including polycystic kidney disease, tuberous sclerosis, in which there are benign tumors, but there are also problems in axonal polarity in neurons, neural tube defects, myoclonic epilepsy, the crash syndrome, which is a form of spastic paraplegia, and several others. Now, so all of these are caused by mutations or defects in these polarity determinants. But for the remainder of my talk, which Win told me should be of about 40 minutes, otherwise he says he's going to pull a hook, and because he has to leave. Anyway, so for the remainder of the talk, I'm going to discuss the trafficking machinery and the cytoskeleton that underlies polarized sorting in neurons, and the defects in these determinants that are responsible for some diseases. Okay, on to neurons. As I mentioned before, neurons are polarized cells. They have dendrites and they have axons. And dendrites receive the nerve impulse from other cells, and axons convey the action potential, the signals, onto other neurons or to muscle cells at the neuromuscular junction. Now, these specialized functions of dendrites and axons are underlined by a different composition of the plasma membrane. Dendrites have neurotransmitter receptors, for instance, glutamate receptors, whereas axons have neurotransmitter transporters that package those neurotransmitters into synaptic vesicles. And they also have different types of adhesion molecules. For instance, neuroligin 1 in the dendrites and neurexin in the axon that interact with each other to form synapses where synaptic transmission occurs. So one fundamental question then in cell polarity is, how is this polarized distribution of proteins, plasma membrane proteins, achieved? Because all of these proteins are encoded in the DNA, they are transcribed into messenger RNA, they are translated in association with the rough endoplasmic reticulum. They're all together at some point. And yet some of them accumulate in the dendrites and others accumulate in the axon. Now, the mechanisms of polarized sorting are complex, and I will not be able to address the multiple factors that contribute to this polarized distribution of proteins. But one important factor is the packaging of these 2 types of protein into different transport carriers in the soma of the dendrites. Although they in the soma of the neurons, although they are all made together in the endoplasmic reticulum, and they all travel together through the Golgi complex. At some point, these two groups of proteins are sorted into different transport carriers, dendritic carriers and axonal carriers. So how does that sorting occur? But before I address that question, I would like to show you a little movie made by one of my postdoctoral fellows, which shows a rat hippocampal neuron, which is a model system that we use for many of our studies. And this is the soma of the neuron, and these are the dendrites. And in red is the transferrin receptor, which, as you can see, is a somatodendritic protein, but is not found in the axon, which is labeled in green. In contrast, the adhesion molecule NGCAM is an axonal protein and is labeled with green here, and is, for the most part, not found in the soma and the dendrites. So here you can see how you can visualize the polarized distribution of two types of protein in neurons. One is axonal, and the other is somatodendritic. Now, if we now do live cell imaging, this is a live neuron, and we are going to photobleach here, and then you are going to see the vesicles entering the axon and entering the neighboring dendrite. And you will be able to appreciate, now we will photobleach that green goes into the green, into the axon, and red goes into red, into the dendrite, for the most part. I hope you can appreciate that with this lighting. So you can see sorting now in play. So emanating from the soma, some carriers go into the axon, while others go into the dendrites. So sorting has occurred in the soma of the neuron. So how does that sorting occur? How are these two types of protein packaged into different carriers? And here I have to talk about our work, because this is a question that we began addressing about 4 or 5 years ago now. And we hypothesized that by analogy to other sorting events that we had studied in other cells, that this sorting likely involved sorting signals in the transmembrane proteins. So axonal proteins and dendritic proteins have intrinsic signals that told them where to go, and that those signals would be recognized by adapter proteins that are components of protein coats, and that this recognition would lead to the packaging of those proteins into either a dendritically targeted vesicle or an axonally targeted vesicle. So that was a hypothesis that we set out to test. And we did it for many different plasma membrane proteins. And I'm just going to show you one example of the work that we have done, using as a model protein the copper transporting ATPase, ATP7B. So this is a multi-spanning membrane protein. It crosses the membrane many times, and its role is to transport copper across membranes. Now, this protein is expressed mainly in the liver, but it's also expressed in smaller amounts in the kidneys and in the brain, but not in other tissues. So it has a rather restricted pattern of expression. And mutations in this copper transporting ATPase are the cause. Okay, so this is what it does. It loads copper into copper-dependent enzymes. And mutations in this protein cause Wilson's disease, which is a disease in which copper accumulates in the liver, causing liver disease. But there are also neurological and psychiatric symptoms that could either be secondary to the liver dysfunction, or they could have a primary origin because of the presence of this protein in the brain as well. Okay, so this is the protein, ATP7B. So one thing that we do in our work is we take these rat hippocampal neurons in primary culture and we transfect them with ATP7B tagged with the green fluorescent protein. And this is what you see. These are such beautiful cells. This is a neuron, and here is the soma. These are the dendrites. And here, labeled with red arrowheads, that's the axon. And as you can see, this protein is mainly found in the soma and the dendrites, but there is very little in the axon. It's polarized to the somatodendritic domain. It doesn't mean that there is nothing in the axon. There is a little bit, but the ratio of dendrite to axon intensity is approximately tenfold. It's a somatodendritic protein. Now, this protein has a cytosolic tail, and in that tail, there are two leucine residues that we like to refer to as a dileucine signal. And when we mutate those two leucine residues to two alanine residues, and we express the mutant protein, this is what happens. Now, the protein is not only in the soma and the dendrites, but now it appears in the axon, which is very long and highly branched. So I hope you can appreciate the striking contrast between these two images. And this is just by mutating two amino acids in a protein that has more than 1,000 amino acids. So this dileucine, therefore, is part of this hypothetical sorting signal for somatodendritic sorting, because when you mutate it, the protein loses its polarity, and it's both in the dendrite and the axon. For this protein to be restricted to the somatodendritic domain, it needs to have those two leucine residues. That's part of the signal. Now, we hypothesized that that signal would be recognized by an adapter protein. And over the years in my lab, we have studied many different adapter proteins that recognize sorting signals. And in particular, there is a family that recognizes dileucine signals. We already knew that. And we focused on one of these adapter protein complexes named as AP1 as a candidate for the recognition of that dileucine signal from ATP7B, because it was already known to bind dileucine signals in other contexts. And also because we had done previous work implicating this AP1 complex in polarized sorting in epithelial cells, in sorting to the basolateral domain. So we suspected that this could also be involved in polarized sorting in neurons. This complex has 4 subunits named gamma, beta 1, mu 1, and sigma 1. Several of these subunits occur as isoforms encoded by different genes. And it's a clathrin-associated protein. Clathrin is a protein that forms this coat around vesicles, and it localizes to the trans-Golgi network, which in neurons is found in the soma of the cell, and that's where the sorting generally occurs. So in order to test whether AP1 was responsible for the recognition of that dileucine signal in ATP7B, we used a dominant negative approach. So we again used rat hippocampal neurons. This is again ATP7B. It's in the soma and the dendrites, very little in the axon. And when we overexpress the sigma 1 subunit of AP1, the normal sigma 1 subunit, which is the one that recognizes dileucine signals, nothing happened. ATP7B was still somatodendritic. But if we overexpress the sigma 1 mutant that was unable to recognize the dileucine signals, it had a dominant negative effect, and ATP7B now appeared in the axon. So this was then proof that an interaction between this dileucine signal in the tail of ATP7B and the AP1 complex was responsible for the packaging of this protein into somatodendritic transport carriers for restriction of this protein to the somatodendritic domain and away from the axon. So AP1, therefore, functions in the soma of the neuron to recognize a dileucine signal in the case of ATP7B for transport to the dendrites. If you either mutate the signal or interfere with the AP1 complex, now ATP7B is no longer restricted to the dendrites and it appears in the axon. Okay, so in addition, our colleague from NICHD, Steve Kaler, also found that a ubiquitous form of the copper transporter ATPase, ATP7A, also behaves in a similar manner, and is sorted to the somatodendritic domain of neurons by virtue of interactions between a dileucin signal and the AP1 adapter complex. And we also demonstrated the same for various other cargos, other plasma membrane proteins that go to the dendrites. So AP1 is really a global regulator of polarized sorting in neurons, as well as in epithelial cells. And it functions to make sure that a subset of plasma membrane proteins go to one domain, the somatodendritic or the basolateral domain, and not to the other domain, the axonal or the apical domain. Now, okay, so does this have any relevance to disease, which is why I think many of you are attending this course? Well, yes, indeed, and we are now beginning to, the connections are now beginning to emerge as some diseases are identified in which there is a mutation in some components of this sorting machinery. In particular, mutations in each of the three isoforms of the sigma-1 subunit of AP1, sigma-1a, sigma-1b, and sigma-1c, have now been shown to be the cause of a different heritable disorder. So these diseases are known as Mednick syndrome, which is due to mutations in sigma-1a, Fried-Pettigrew syndrome, which is due to mutations in sigma-1b, and pustular psoriasis, which is due to mutations in sigma-1c. So mutations in each of three isoforms of an AP1 subunit causes a different genetic disorder. Now, interestingly, two of them, Mednick and Fried-Pettigrew, present with mental retardation and other neurological abnormalities. So it's mainly a disease of the central nervous system. But in addition, two of them, Mednick and pustular psoriasis, present with defects in cells, tissues, or organs that have polarized cells within them. For instance, the intestine, there is enteropathy in the case of Mednick syndrome. The inner ear, where there are hair cells that are polarized as well, because there is deafness, and problems in the skin, and the same in pustular psoriasis. So I think that the correlation between the symptoms that are seen in these AP1 deficiency disorders and the role of AP1 in polarized cells are really striking and suggest that these diseases result from failure of polarized sorting of some cargos, some plasma membrane proteins into both neurons and polarized epithelial cells. And I would like to tell you a little bit more about the Mednick syndrome, where that connection may be a little bit clearer. So this is a Mednick patient that was seen here at the clinical center in Building 10 by Steve Kaler, and he kindly provided me with these pictures. And what you will be able to appreciate in this patient is mainly the skin symptoms. There is rash and there is keratodermia, and so the baby has a hearing aid because of the deafness. But you cannot see the other symptoms. Now, what are those other symptoms? Interestingly, Mednick is not the name of the physician who described this disease. It's an acronym for the main symptoms, which are mental retardation, enteropathy, deafness, neuropathy, ichthyosis, and keratodermia, Mednick. Now, interestingly, Diego Martinelli, who is now a postdoctoral fellow in Steve Kaler's lab, in his previous lab had found that in addition to these problems, these patients also had hepatomegaly, cholestasis, low serum and CSF copper, high liver copper, and other defects of liver and copper metabolism that are reminiscent mainly of Wilson's disease, which, as I mentioned before, is due to mutations in ATP7B. So here you can see the very strong correlation between mutation in ATP7B and the adapter protein that recognizes the dilucine signal in ATP7B. So if you have a mutation in either the copper transporter signal or the adapter that sorts it, you have some of the symptoms are similar. Now, these patients, unfortunately, have a lot of other problems. And those are probably due to the missorting of other plasma membrane proteins that are also dependent on AP1. And we are now interested in identifying what those other plasma membrane proteins that are AP1 dependent are, because then you may be able to help these patients with some therapies. In the case of the copper metabolism defects in these patients, they are treated with zinc acetate, which is a standard therapy for Wilson's disease. It decreases copper uptake in the intestine. And the same therapy can be used for these patients, and actually their liver function and their copper metabolism improves quite significantly. And with it, a lot of secondary problems also improve. So if we could identify those other AP1 dependent cargos, then there might be other things that we can do for these patients. Okay, so I'm thinking of the hook. Okay, so now, so this is about AP1 dependent sorting. However, there is another adaptor protein complex which is related to AP1 and which is known as AP4, which has also been shown to play a role in polarized sorting in neurons. But it sorts a different subset of proteins to the somatodendritic domain. It sorts a different type of glutamate receptors. AMPA type of glutamate receptors are dependent on AP4. And it functions similarly to AP1 by recognizing signals in the tail of these receptors, probably at the trans-Golgi network, for sorting to the somatodendritic domain. And mutations in AP4 then result in missorting of these receptors to the axon in a similar manner to the defective expression of AP1. Now, interestingly, other diseases have been associated with AP4 defects. AP4 has 4 subunits, epsilon, beta 4, mu 4, and sigma 4, which are homologous to those of AP1. And mutations in each of the 4 subunits of AP4 have now been shown to be the cause of hereditary spastic paraplegia. These types, 47, 50, 51, and 52. So this is a movement disorder. There is initially hypotonia that later develops into hypertonia. There is also mental retardation. And mutations in each of the 4 subunits have been now found in patients, and they all have the same manifestations. And also, Dennis Reina, who's here in the audience, recently identified variations in the epsilon subunit of AP4 in heterozygous patients that have non-syndromic developmental stuttering. So the homozygous mutations result in these very severe spastic paraplegia and mental retardation. These heterozygous variations in the epsilon subunit are associated with familial stuttering. So here again, you can see that another adaptor protein complex involved in polarized sorting in neurons is defective in diseases that mainly compromise the function of the central nervous system. So we think that these diseases are also going to be defects in polarized sorting. Okay, so AP1 and AP4 function in sorting to the somatodendritic domain. Now, so far I've been telling you about sorting into this type of carrier, into the somatodendritic carrier. How about the axonal carriers? How are proteins sorted to the axon? And unfortunately, I don't have much to offer you because it's not known very well. Believe it or not, some of you may be surprised that we do not understand very well how proteins are packaged into axonal transport carriers. What are the signals? What are the adapters that recognize those signals? Still not very well understood, and I will leave it at that. So I think it's a very exciting goal of future research. Now, I would like to now, in the next 5 or 10 minutes, like to address a question. So now I've told you how proteins are packaged into two different types of carriers. Here we understand the mechanism. Here we don't understand it very well, but they are packaged into two types of vesicular transport carriers. But now there is an additional problem in polarity. How are these carriers, the carriers themselves, now targeted to the dendritic or to the axonal domain? Why don't these carriers go to the dendrites? And why don't these go to the axon, right? And you saw in the movie that green went into green and red went into red. They are being sorted. How do they know where to go? And that's a question that I would like to very briefly address now. And before I address that question, I would like to ask this question. What is the boundary between the axonal and somatodendritic domains? Wynne already alluded to this in his introduction, that there must be a boundary that separates the two domains. In epithelial cells are the cell-cell junctions. The tight junctions separate the apical from the basal domain. Where is that boundary in the neurons? Well, for plasma membrane proteins, that boundary is in the so-called axon initial segment, or AIS. So it's an area in the first part of the axon. And this axon initial segment is a very specialized structure in the neuron, which has a very high concentration of voltage-gated channels, sodium and potassium channels, which are responsible for the generation of the action potential. So this is where the neuron receives all the information coming from the dendrites and from the soma. And here is where the decision is made. Okay, I have all this information. I'll fire an action potential that will then be propagated down the axon for transmission to other cells. So this is really a functionally critical structure in the neuron. So that's the main role of the axon initial segment. But it has an additional role. It functions as a barrier for the lateral diffusion of the axonal plasma membrane proteins and the somatodendritic plasma membrane proteins. They cannot diffuse through this densely packed cluster of voltage-gated channels and adhesion molecules and other things. So that is the boundary for the axonal and somatodendritic plasma membrane proteins. Now, interestingly, the axon initial segment has also been proposed to function as a filter, an actin-based filter that somehow allows the axonal transport carriers to go through it unimpeded into the axon, but blocks the transport of dendritic carriers. So we were intrigued by that mechanism, and for a while we wanted to study it. What kind of a filter is it? How does it work? But we did some experiments that actually persuaded us that the axon initial segment was not the boundary for cytoplasmic organelles. It's a boundary for plasma membrane proteins, but not for cytoplasmic organelles. Why? First of all, because we couldn't find a dense network of actin filaments in the axon initial segment. Yeah, there was some actin, but it was not what you would expect for a filter. And then other people actually did it much better than we did it, and they demonstrated by very sophisticated techniques that there is no actin filter in the cytoplasm of the axon initial segment. There is no plug, there is no filter there. And second, we observed that that indeed axonal transport carriers were able to go through this region of the neuron without any problem. But the dendritic carriers were unable to enter the axon, not at the axon initial segment, but at an earlier region in what's called the axon hillock. This area of the neuron is referred to as the axon hillock. And we observed many instances, and I don't have time to show you this, of dendritic carriers that tried to enter this region before the axon initial segment, and they were turned back to the soma. Others came and tried to enter that region, and they were turned back. They were unable to enter. And I will show you just one example of the many experiments that we did. This is from our work published in 2015. This is a transferrin receptor, a somatodendritic protein. This is the soma, these are the dendrites, and this is the axon initial segment labeled with an antibody to ankyrin G. And so look at where the boundary is, where the transferrin receptor stops. Here, before the axon initial segment, at higher magnification, there is an empty area, there is a void where the transferrin receptor cannot enter. So this is where the sorting of dendritic and axonal carriers is taking place. And we name this region the pre-axonal exclusion zone, or PAZE. Now we said, well, this is interesting, but nobody saw this before. So we went to old literature, right? We went to this paper from 1993 in which they had stained neurons with germ agglutinin, which labels sugars in endosomes and all that. And they had noticed that there was this sharp boundary between the soma and the axon hillock. Look at this. So there is this empty area. So that's because endosomes couldn't get into that area. And we then went even further back. 1895, Hans Held published these beautiful drawings. Hans Held, the guy who identified the calyx of Held structure in a particular type of neuron. And look at this. This is a rough endoplasmic reticulum stained with a Nissl stain. And look here, there is that empty area. This is another drawing by Held. There you see this empty area. So the pre-axonal exclusion zone was always there. Its functional significance had not been appreciated at the time. So we now know that this pre-axonal exclusion zone is a region that organelles that are going to enter the axon can go through without any problem, but organelles that are going to be restricted to the somatodendritic domain cannot penetrate. So what is the mechanism? And I don't have time to go into this, but we went back to an older idea that sorting of axonal and somatodendritic carriers or organelles is dictated by the microtubule motors that can interact with those organelles, with those carriers. For instance, axonal carriers will recruit axonal kinesins, which are microtubule motors that have an affinity for axonal microtubules. And that's how they will be able to be transported along the axon. Whereas somatodendritic organelles will bind a different type of kinesin, a different type of microtubule motor, or dynein, another type of microtubule motor. And that interaction will be responsible for the transport to the dendrites. So in our view, there is no physical filter. What there is a different association with different subsets of microtubule motors, some that mediate transport to the axon and some that mediate transport to the dendrites. So how is that possible? Well, there are about 45 kinesins encoded in our genomes, lots of them. These are microtubule motors that move all sorts of things within cells. And in neurons, some of them, most of them actually, are axonal, but there are a few of them that are dendritic. And that's how, if an organelle associates with one or the other, then it will tend to be transported to the axon or to the dendrites. And how do those kinesins know what microtubules to associate with, axonal versus dendritic microtubules? Well, first of all, axonal and dendritic microtubules have different properties. They have different polarity, and I don't have time to get into that. But they also have different post-translational modifications of tubulin, acetylation, tyrosination, polyglutamination, and others, or different microtubule-associated proteins. So these kinesins are smart motors. And I didn't coin that phrase, somebody else did. They are smart. They can recognize these different microtubules, and they know whether to associate with an axonal microtubule or with a dendritic microtubule. And then there are adapter proteins that will then couple the organelles to those smart microtubule motors, and then they will be taken to the axon or to the dendrite. So this is, I think, the latest view of how these two types of carriers are sorted. Now, before I said that there were mutations in adapters and code proteins that caused some neurological diseases. Of course, there are also mutations in components of these cytoskeletal motors that are responsible for various diseases, actually a very good number of them. And I'm just going to mention some of them so that you can appreciate the importance of this process of targeting axonal and dendritic carriers to their corresponding domains. First of all, there are other forms of spastic paraplegia. So before I told you that mutations in the subunits of AP4 cause a form of spastic paraplegia, a movement disorder with mental retardation. Well, mutations in a particular kinesin, which is an axonal kinesin, causes a different type of spastic paraplegia, SPG10. Then there are mutations in another kinesin, KIF1A, that causes also spastic paraplegia. This is another axonal kinesin. There are mutations in yet another kinesin that cause another form of cortical dysplasia with brain malformations, type 2. There are also changes in the expression levels of a kinesin light chain, which associates with these two kinesins, KIF5N and KIF5C, that causes yet another form of spastic paraplegia. And finally, there is something called Spohn syndrome, which is another form of spastic paraplegia. And finally, there are the tauopathies. Tau is a microtubule-associated protein, and mutations in tau underlie familiar forms of frontotemporal dementia. And also there are common there is common pathology of tau in diseases such as Alzheimer's disease as well, the neurofibrillary tangles that you can see in Alzheimer's disease. So, and with this, I would like to summarize what I've presented, that polarized sorting then depends on two selective processes. The first one is packaging the proteins into at least two types of vesicles that will go to the somatodendritic or to the axonal domains. And then the second process is the delivery of those, the accurate delivery of those cargoes to the corresponding domains. You first package and then you deliver them. So the first process is mediated by signal adapter interactions. That's how they get packaged. And the second process is mediated by interaction of those carriers with microtubule motors and with the underlying microtubule cytoskeleton. And defects in either the packaging or the targeting of the carriers is a relatively common cause of neurological disease, certainly inherited forms of neurological disease, but most likely also in other spontaneous diseases, but that is less well understood. And I also wanted to mention the fact that that most recent studies suggest that whereas that the boundary for the somatodendritic and axonal domains at the plasma membrane is at the axon initial segment, but in the cytoplasm, it's in this pre-axonal exclusion zone, and they operate by different mechanisms. And with this, I thank you very much for your attention, and I hope we'll have time for some questions if you have any.
That was fantastic, and we have time for some questions if you wish. Or you want to hold on until the discussion afterwards?
Thank you. My question is more on the signal transduction between the two different carriers, could you give some share some thoughts on it as to even for a case where it is related to a disease, is it similar to normal state, or there is difference between signal transduction?
What do you mean signal transduction? You mean in terms of the sorting signals or kinases and
Sorting
proteins?
between
For the sorting
dendritic,
signals,
yeah.
you know, before becoming interested in neurons, I was a cell biologist working with epithelial cells and other cell types. And going into neurobiology, I figured, well, a lot of these things must be known. Let's see what we can add to it. And then as we started going to the literature, we realized that a lot of very fundamental processes were not very well understood. So our current state of understanding is that we know a lot about somatodendritic sorting, the signals, the dilucin signals, and the adapters, AP1, AP4. We know a lot about that, but we know very little about axonal sorting signals. And that's something that we are interested in, and I'm sure a lot of other people are interested in. So at present, I cannot really tell you how the two mechanisms compare because we know very little about axonal sorting.
Okay.
We do not even know whether axonal sorting will depend on the recognition of a signal by an adapter protein. There could be an entirely different mechanism, which could be lipid-mediated. It could be very different. But anyway, other questions? Yes.
Yeah.
How the neuron knows which part, this part will come develop into axon, this part will in dendrite?
Yeah, so all those determinants that I talked about at the beginning are probably important. So when you work with rat hippocampal neurons in primary culture, you have to isolate the hippocampus from the brain, and then you have to treat it with enzymes to separate all the neurons. Then you put them in culture, and they are all round. They don't have axons, they don't have dendrites, it's just a round cell. And then they attach and they begin to flatten, and then they emit neurites. And then one of them will become the axon. So there a decision was made that one of them will become the axon. And very early on, you have all those polarity determinants that I was talking about, the PAR proteins and crumbs and scribble, and those play a role in determining which of the neurites will become an axon. And then you start polarizing the cytoskeleton. Before you have an axon initial segment or anything, the axonal kinesins have redirected towards one of the neurites. And that's going to become the axon. And then they begin directing a lot of cargoes into that neurite that becomes the axon. So at some point in that round cell, a decision will be made, and one projection will be labeled as the axon, and then that will grow. Yes.
Do you think that model that you outlined for the segregation, where it knows to not send certain things to the axon, do you think that's similar or related to others, to cell differentiation or like yeast daughter cell budding? Do you think this systems might be similar?
Yeah. A lot of these mechanisms of cell polarity are conserved among different polarized cell types. I mentioned epithelial cells and neurons. I could mention endothelial cells and other polarized cells. So they are quite conserved. And they're also quite conserved in other organisms, including yeast. Now, Saccharomyces cerevisiae is a little bit different from other yeasts in that it relies more on the actin cytoskeleton to move organelles rather than the microtubule cytoskeleton. So yeah, with certain adaptations, the mechanisms are quite similar. But Saccharomyces is a little bit different. Other yeasts Fungi, they use more the microtubule cytoskeleton. They are more like a neuron. Saccharomyces is very special in that it uses the actin cytoskeleton, but a lot of these mechanisms are conserved. Yeast has AP1, for instance, and it uses it for polarized sorting in the same way that our neurons.
Okay.
Other questions? Okay, thank you very much.
Okay. All right, thank you very much, Juan. Now, the liver participates in the concept of polarity from many different perspectives, and yet there are many similarities. But I must confess, none of them as elegantly demonstrated as you just saw. But let's follow through here. So hepatocyte polarization has three features to it, which are frankly not well recognized by people who work in the liver field, as distinguished perhaps from the cell biology field. And that is that the mechanisms are complex. They are essential. The absence of polarity is a signal for death in the hepatocyte. And they are virtually neglected in the clinical practice of liver disease. And I'd like to point out to you in the presentation why the basis of these three things. And then I'd like to end with something which I will describe, but you'll be able to see due to my error, which is the introduction of high-resolution study of hepatocyte polarization in living mice. And this is work done by Natalie Porach-Leome, who's sitting right over here, and we'll get to it. Now, for those who don't reside intellectually within the liver, I have to show this slide to point out just the ballpark. Blood, arterial and venous blood, enters on the left-hand side over there. Okay, not my day. On the left-hand side over there, and then passes through the red area, which is the hepatic sinusoid. And that is bathing, for the main part, two rows of hepatocytes, which are here represented as cuboidal cells. In this diagram, they look all alike, and histologically they look alike, but they're carrying out very different functions in different positions along the lobule. Oh, thank you very much, Juan. Yeah, and then between, the blood is running from here to here and then back up to the heart, and between these rows in the opposite direction is the biliary secretion by these hepatocytes. So they are secreting lipids, particularly phospholipid, a detergent, bile acid, which we're going to dwell on a bit. It's the major mechanism by which we eliminate cholesterol, and traditionally has been described as emulsifying fat in the intestinal tract, facilitating its absorption. But in recent years, as I'll show you, it turns out that bile acids are also signaling molecules which play a part in the polarization machinery. And then that secretion into the bile is a major route for the elimination of a wide variety of anionic and cationic organic molecules, drugs, metabolites, some hormones, and so forth. Now, you have to keep in mind that the hepatocytes, they're very different from neurons, and they're very different from the schematized epithelial cells, such as in the intestine, because they have a unique polarization. And one hepatocyte can have up to 3 apical domains. So here's a hepatocyte, here's one bile canaliculus, and that would be another one, and there could be a third one, 1, 2, and 3. So a single cell can have 3 polarized domains. Now this is in complete comparison to columnar epithelial, like in the intestinal tract, where these cells are lined up. There are tight junctions that separate them, but they have a brush border on a single surface that faces into the intestinal lumen. Here, this bile canaliculus is formed by the union of two cells, forming a tight junction, and then delineating a small capillary-like structure, which is the bile canaliculus. And this is what it looks like in scanning electron microscopy. Here would be two hepatocytes. And here's this capillary-like structure with its microvilli. And it's across that plasma membrane that all the secretion into the bile occurs. And this membrane is separated from the basolateral by tight junctional and other complexes. Now, these pictures are misleading. Here in scanning EM, you get one picture. If one were to do histochemical staining of a ATP transporter, the ABC transporter that's found in the canaliculus, it looks sort of like chicken wire in a section. But in reality, what the canaliculus is, that is this biliary system, is a network, as revealed here in a rather elegant cast that was made by Japanese colleagues years ago. And it's this network that has never been reproduced in any culture system except one that I'll show you, and has handicapped our knowledge about polarization mechanisms. Now, the loss of this network results in the inability to secrete, particularly the detergent, the bile acid, and it results in liver failure, cholestasis, a pathology term meaning that the bile stands still, but it's standing still predominantly in the hepatocytes, where it acts as a detergent and has many different toxic effects. Now, why has this whole business been neglected? And here's the answer. Because this is what the normal liver looks like when it goes to a pathologist, or for that matter, even abnormal liver. They stain it with a stain called hematoxylin and eosin, which is sort of in the traditional workhorse, but it doesn't visualize the bile canaliculus. You would never know it was there. And frankly, I think this is the main reason why virtually no attention has been placed to this critical structure, which is essential for the secretion of many metabolites, and particularly the bile acids. Now, the system that we've used to study this is one that was developed years ago by a group in North Carolina and then kind of discarded. These are primary cultures of rodent or human hepatocytes put in a collagen sandwich. And then they are stained, in this case, with a tight junctional marker, a gluten, and an intrinsic protein, a transporter of the canalicular membrane. There's no cell division here. There's virtually no cell death. And what happens sequentially in a remarkably predictable manner is that this network formation occurs over a period of about 6 days. And this system has proved invaluable in studies of the mechanism by which this polarization occurs. Now, another major discovery that put things in a different direction was the finding that there were 6 ATP binding cassette transporters, which are localized, restricted to this apical membrane. They each couple the hydrolysis of ATP to transport a different substrate. This is the one we'll talk about mainly because B11 transports a whole variety of bile acids, the detergents, from the cell into the canaliculus. This one is flipping phosphatidylcholine. This is a sterol transporter. This is a bunch of organic anions, glucuronides, sulfates, and so forth. And each one of these has been associated with an inheritable disease characterized by a phenotype that's the direct result of the failure of these proteins to either be made properly or to wind up in their appropriate localization. Now, this is what we would call functional polarity, as distinguished from what we'll talk about a little later, of structural polarity. Structural meaning that the basic structure of this apical domain is altered. Functional meaning that there are individual components that are affected. So let me tell you a little bit about this one. With the discovery of ABCB11, many people theorized what would a disease look like if it were unable to secrete bile acids into the intestine. And it was all agreed that children who had this would be very sick, and indeed they are. There's a varied phenotype, but they're all characterized by retention of bile acids. And this transporter, there are now over 100 mutations of a variety of molecular types, basically either isn't made or isn't processed properly, misfolded, or isn't trafficked properly into the apical membrane. And there are mutations affecting all of these. The net effect is severe liver damage in a child, which progresses to scarring and cirrhosis. And the only cure is transplantation. This gene is only expressed in the hepatocyte. So this is a kind of proof of principle that if the cell is unable to secrete the detergent, bad things happen. We don't know what. We'll talk about that in a minute. But there are many other situations that are not clearly inheritable where the same phenotype occurs, and that's very common in hepatology. So we became interested in the question about how does this transporter and the other ones get from the trans Golgi to the apical membrane. Other proteins that reside within this apical membrane are trafficked from the Golgi to the basolateral domain and then transcytosed into this apical domain. But we discovered that the ATP binding cassette transporters do not go by that indirect route. They actually are trafficked directly. Furthermore, they exist in a large intracellular pool, a recycling endosome pool, where there are 10 times as much of these receptors in this recycling pool as there actually are in the membrane. Physiologically, this movement from the pool to the membrane is stimulated by things like eating, because then bile acids return to the liver, and bile acids are a stimulus for this movement from the pool. Also, various peptide hormones that activate adenyl cyclase do the same thing. So this large pool accommodates the wave of bile acids coming back after a meal, moves to the membrane, facilitates its secretion, and then it recycles back. Now, I can't show you the movie, my mistake, but what this movie would show you, this is ABCB11, and it is in the microtubular organizing center, and it actually moves as long tubular vesicles, which proceed to specific sites in the apical membrane. And those specific sites turn out to be, from studies done by several laboratories, to be largely a docking protein called syntactin 3. So these vesicles move, they oscillate, and there are smaller vesicles that come in from the side. And all of this is microtubular dependent. It's completely obliterated if a microtubule inhibitor like nicotazole, for example, is added. Now, we then spent the next 10 years trying to identify players that are involved in this movement. And we used a wide variety of techniques, most of them biochemical, yeast to hybrid screens, some knockouts, some silencing RNA, et cetera. And so our current understanding of the process is that these transporters are made, they're glycosylated, N-glycosylated in the Golgi. They traffic along microtubules, and they require the dynamic portion of the microtubule, the plus N, which is marked by a CLIP 170 and EB10, which turn out to be important. And they enter this large recycling pool, which I described to you, and they move to the apical membrane, docked to syntaxin. This movement is regulated by this complex of a RAB protein, RAB11A, and its adapter, and a myosin motor molecule, which we'll talk about in a moment. Now, the recycling back is influenced by these proteins and is a clathrin-dependent process. And then there also is degradation, where some of this transporter winds up within the cells in a place where it's degraded. Now, some of the things indicated in yellow here, I'll comment about because they turned out to be very important. When we affect these various parameters indicated in yellow, which I'll show you, the effect is not just one on trafficking, the effect is one on polarization. But this is not just functional polarization. It isn't just that the transporter doesn't get from here to here. It's that this is converted into like a planar epithelium. There is structural change. So here's a cartoon of what is believed to be how post-Golgi vesicles here, carrying the cargo, they utilize this Rab11A complex with its adapter protein, which binds to a myosin 5B motor, which then moves it along in this case, the actin filaments. Now, as I'll show you, there's another adapter protein that was discovered called VAMP, which interacts with Rab11, and mutations cause a human disease. In studying this, we generated a Rab11A locked GDP molecule and then transfected it into cells. The question was, if you block Rab11A, or you put a construct of the myosin motor, which is only the tail, not having the motor domain, this is like an inhibitory complex, we thought that if we express these things, we would just be inhibiting trafficking from the recycling thing. But what turned out was, as I anticipated a moment ago for you, we blocked polarization completely. So here, for example, this is in a cell line that polarizes. This is in primary hepatocytes. And it's the same thing. These cells in culture grow, and the increasing number of cells that polarizes get up here. And if we express these constructs, the inhibitory myosin, the GDP-locked Rab11A, and this is a control virus just to show that that's not the effect. What happens is, remarkably enough, the cells don't die, growth continues, but they don't polarize. So here is the polarization. This is in these whip cells. And the control, about 60% are polarized at the time, expressing either the GDP-locked Rab or the myosin motorless domain doesn't just affect trafficking, it's affecting the whole machinery of polarization. So what sort of a cue exists between the recycling pathway and the polarization machinery? So Juan commented about all of these I just mentioned. In the liver, they are the same. These are the hepatocyte components of the whole complex process: cell adhesion molecules, cell junctions, various cytoskeletal factors, the intracellular trafficking mechanism, and it's all dependent on cellular energy. So what we have done through the years is to inhibit components of these trafficking systems, the junction, the recycling, energy, and observed using the imaging techniques, which I quickly illustrated for you, as well as other techniques, to show that not only is ABCB11 trafficking impaired by these maneuvers, but the cells don't polarize, or if they're introduced when they have been polarized in the culture system, they depolarize. So these are required for creation and maintenance. So for example, inhibitory inhibition of microtubules by a variety of means, including an adapter protein Redixin, which was done in a knockout system and with a silencing RNA. With Anamouche, we've done studies with a PAR1 protein involved in junctional formation, her knockout, and a silencing RNA, and I told you about the recycling ones. And we're going to talk for a minute about the role of energy in this entire process. Now, all of these things, all of these manipulations led to the failure to polarize, the inability to traffic, and when done in a polarized cell, rapidly to depolarize. Now, here's a case history, which was presented to me actually when I was visiting in Holland a few years ago. This is an 8-year-old schoolgirl. She had a normal birth, was 6 pounds, but she promptly began to lose weight, was dehydrated, had diarrhea on ingestion of any kind of food, including milk. She was a medical emergency, and she was treated by putting an indwelling line and maintained with parenteral nutrition, which was sustained for a period of 4 years. At about 1 month of age, her jaundice, which was present at birth, became much more severe, and she had increasing liver damage over the next 3 years. An intestinal biopsy, which I'll show you an example of next, was performed, which provided the diagnosis. At age 4, she had a successful transplantation of the entire small intestine and liver, and has been on immunosuppressive drugs ever since. By age 5 or 6, she had regained more or less normal growth and nutrition. I couldn't believe it when they presented this as a case history, and in walked this 8-year-old girl. She had a disease called microvillus inclusion body disease, in which this is the intestine, small intestine from a biopsy. Now, normally the small intestine has the surface of it sort of looks like a picket fence. But all of the surface microvilli are gone. They don't appear. Pathologists were impressed by this inclusion, which looks a little bit like the microvilli of the surface. And so that's why they call it inclusion disease, but it still isn't clear whether this represents a brush border that is inappropriately made, trafficked, or whether it's more readily reabsorbed. That hasn't been studied. But the net effect is that this young lady and about 200 others lack the brush border on the surface. The polarity is lost. This is converted into a planar cell, still having tight junctions, but it can't function in a polarized way. Now, this is recessively inherited, and a similar process occurs in the liver, but we don't have as much detail about it as we did. Now, this is due to a series of mutations, there are 16 described so far, in the myosin 5B gene, first described in this article in Nature Genetics. Similarly, this has been observed in mice where this has been knocked out. So here we see evidence of a profound human disorder affecting liver and kidney due to a disruption of the polarity targeting machinery. Now let's turn for a few minutes about the fact that this process and all of the processes that Juan showed you basically require cellular energy. Now, the two key players that we have been working with are these two serine threonine kinases. LKB1 was initially described as a tumor suppressor. It's mutated in the human cancer disease called the Putz-Jeghers syndrome. So phosphorylation of serine 431 on LKB1 requires the presence of a scaffolding and an adapter protein. Exactly what the agonists are that lead to this phosphorylation, growth factors or anything else, is yet to be really defined. One of the major downstream targets of LKB1 is another threonine kinase, AMPK. AMP-activated protein kinase exists, and there are 14 members of the family, but there's only one which is primarily related to the polarization machinery. And it's activated by LKB1 and also by a rise of AMP in the cell. So it's thought of as the metabolic sensor. When ATP has been consumed and the ratio of AMP goes up, that adds to phosphorylation of this site as well. And so the relationship of AMP kinase to cellular metabolism has been studied extensively. But it wasn't until about 2000, I guess 4 was the first one, in which it was shown in different cell systems that the AMPK-LKB system is required for cellular polarity. If I use this to summarize a little background of what AMPK, how AMPK works and what we find in the liver, I will take you through it. So here we are, the activation of LKB1, which it turns out is regulated in part by bile acid, which we showed, acting through a cyclic AMP mechanism. But that's not the only way in which LKB1 is activated, and as I pointed out, the real agonists for it have not been defined. Phosphorylation of AMPK by either AMP or activation of LKB1 leads to a profound suppression of all systems that utilize ATP within the cell. That is, lipid synthesis is inhibited, protein synthesis, growth. And this is the basis, by the way, of the relationship of AMPK to the autophagy pathways and to mTOR and its relationship to cancer. Now, the one system that is activated is cellular catabolism, and we find that what's activated is cellular polarization. So I will show you that AMPK activation leads to enhancement of trafficking, tight junction formation, and cytoskeletal organization. While doing these studies in these cell cultures in Jennifer Lippincott-Schwartz's lab, where many people were working on mitochondria, we were asked, what do the mitochondria look like? And we had a huge surprise. When these cells are isolated on day 1, the mitochondria, stained here, are all very small. There's little evidence of their degradation, mitophagy, but they are all very, very small, and here it's quantified. But then, very rapidly, over a period of a couple of days, the mitochondria undergo fusion, and they become like giant mitochondria, extending throughout the cell. Now, fission and fusion are regulated by complexes of proteins, some of which are indicated here. Fusion is accentuated by expression of MFN1 and 2, which are outer mitochondrial membrane proteins, and also by OPA1, which is an inner mitochondrial protein. The fission machinery is largely driven by a complex which is getting more complicated every day, involving the protein DRP1, which accentuates this part of the process and tends to lead to the breakdown of mitochondria with autophagy and their turnover, and then activation of biogenesis and also of stimulation to form these long, elongated mitochondria. So Dong Fu, when he was in our laboratory, measured some of these proteins and discovered that the process of change from mitochondrial fission, breaking down, to fusion was associated with activation of MFN1 and OPA here. No change in structural proteins, indicating this was not a generalized effect, and little demonstrable alteration in the DPN fission process. So something is happening to activate as a result of the stress of cellular isolation to activate these cells. Now, what happens when the mitochondria become fused? Well, their potential is driven upward. Their ATP synthesis, which is very low, is restored, and they're much more efficient in making ATP when they're fused than when they're not. And interestingly enough, the initial ATP that's made for the first 3 days is derived from oxidative phosphorylation, as shown here by the fact that that ATP is inhibited by oligomycin. Whereas it's only after day 3 that apparently glycolysis kicks in, and we see inhibition of AMP kinase by 2-deoxyglucose. Now, what does this mean? What it suggests is that after stress, the mitochondria are responding preferentially by fusing using oxidative generating ATP by oxidative phosphorylation, and that this is an initial stress response. Well, how does AMPK affect mitochondria? The traditional and classic studies by Hardy show that it acts indirectly by increasing glucose uptake, affecting the glucose transporter and enhancing glycolysis. And then Arias showed that as a product of metabolism, NAD activates sirtuin, which then deacetylates PGC1 alpha, leading to mitochondrial biogenesis. But more recently, direct effects of AMPK on mitochondria were shown by Bruce Spiegelman, who showed that AMPK can directly phosphorylate this transcription factor, which leads to new mitochondria. And more recently, Ruben Shaw showed that AMPK can directly interact with the mitochondrial fusion apparatus itself. The conclusion of this is that AMPK is a major regulator of mitochondrial dynamics and metabolism. Now, the bile acid physiologically plays a part in facilitating the polarized phenotype. But whenever bile acid accumulates within the cell, it leads to this mitochondrial stress reaction that I mentioned, and it leads to fragmentation, reduced beta oxidation, increased reactive oxygen, and ultimately other organelle toxicity. The important point is that bile acid accumulation is the common pathway for intracellular injury, and that leads to loss of polarity, and loss of polarity is the beginning of cell death. Now, this schema has arrows, but the arrows should have question marks because we don't know all of the processes that are involved. But it's clear that bile acids have some regulatory role in polarization, that the 2 kinases are critical for ATP generation and maintenance, and that they are acting directly and indirectly through mitochondrial fission, fusion, and other events. But whether AMPK is only acting in this way, or whether it cannot be acting on downstream targets in the polarization machinery itself, has yet to be worked out. Many of the components of the polarization machinery have AMPK consensus sites, but the only one where functional polarization has been shown to be important is work by Anna Musch and Einstein, who showed that the PAR1 protein must be phosphorylated in order to maintain its tight junctional formation. Now, there are a variety of inherited hepatocyte polarity diseases. The ones on your left are the ones I referred to before as functional. So these are the transporter disease in which the defect resides either in transcription and misfolding or in some instances in altered trafficking. On the right are a series of disorders in which mutations exist in the structural machinery. And I showed you one, myosin 5B. I'll show you another now in the VPAR VPS33B complex, which interacts with this. There are disorders in which tight junctional protein 2 and claudin are mutated with having clinical consequences in the production of liver disease, particularly in children. And there is also a recently described mutation in syntactin 3, the docking protein, which has a phenotype virtually identical to that seen in the microvillar inclusion body disease. So mutations in VPAR cause this syndrome. There are contractures of the joint, there's kidney dysfunction, and there's impaired biliary secretion and cholestasis. And there are defects in epithelial polarization. So this work was done by Paul Gieson, with whom we collaborated. Paul is in London. And one initial finding was that the bile acid transporter, instead of normally being seen in this chicken wire-like localization, is actually in the basolateral domain. It's misfolded, it's misdirected. And here's a cartoon that indicates the summation of all of the studies that were done. So under normal conditions, the ABC transporter on the apical membrane goes through the apical cycling pool, and a portion of it goes through the endosomal pool and is eventually degraded. But if VPAR is mutated, and this has been done experimentally in mice, and there are 8 different mutations that have been described in children so far, what happens is that the transporters do not remain on the surface of the apical membrane for a sufficient period of time. They are trafficked normally there, but they are misdirected to the basolateral domain, and a portion go into an intracellular domain that we have yet to identify. Now, VPAR interacts with the RAB11A myosin 5B complex. Exactly how it regulates it isn't known. When it's defective, we get mislocalization of the transporters and an accelerated loss from the apical membrane. So we propose that VPAR regulates the rate of recycling and degradation of membrane proteins. And studies are going on regarding its role in intracellular trafficking. Now, these are rare inheritable diseases, but in the same way as Juan demonstrated, the studies of the rare inheritable neurologic diseases provide key information as to the function of normal pathways. The same thing is true in the liver, but we have a much longer way to go. On the other hand, the real interest down the line is what happens in acquired disease, which is much more complex. Now, one example of this are drugs. There are a variety of drugs that are toxic to the liver, some more severe than others, and a great deal of interest in trying to delineate toxicity before it is given to patients and sold commercially. And attention has become focused on the mitochondria in most of these processes for some of the reasons that I showed you a moment ago. And recently, our colleague Don Fu, now in Australia, has proposed and done some very interesting studies with several hepatotoxic drugs, which lead to impairment of fusion rapidly to mitochondrial fission, to autophagy, mitochondrial damage, and so forth. And if one restores the AMPK to the system, it enhances biogenesis, fusion, and prevents and to some extent reverses the toxicity with some drugs. Well, this is the beginning of a series of studies to relate energy metabolism, polarity, mitochondrial biology, if you will, and drug toxicity. So the same thing applies with acquired diseases like viral hepatitis. Hepatitis C requires attachment to claudin 1 for entry into the cell. What is the role of either mutations or of toxicity in that process in the disease? And there are many other examples. Now I'll conclude with these studies of what happens now if you have a selective liver-specific LKB1 deficiency. And so this was generated in our lab when Laszlo Homolya was with us a couple of years ago. This is an albumin-Cre knockout, and the Cre is expressed at about day 6 or 7, at which time then the LKB1 knockout is expressed. And so the animals from that point on, they lose weight, they exhibit a pretty severe phenotype, they're jaundiced, and they die within 28 to 30 days. Now, the interesting thing is the liver does not show signs of inflammation or necrosis. So this is a functional disorder. Michael Jarnick has been doing electron microscopic studies of these animals with us for a couple of years now, and came up with a fascinating observation. Here is the control. At birth or day, week, the biocanalicus is discernible, and that's within the normal range. Here at 4 weeks, these are microvilli. These are tight junctions, the heavy osmophilic staining, and this is just exaggerated from here. But in contrast, in the LKB1 knockout, in the first week, looks perfectly normal because the transgenic has not been expressed. But by 4 weeks, a remarkable finding. With some consistency, there are markedly reduced numbers of biocanaliculi to begin with, and when they're present, they tend to show the fact they are not restricted just to this domain. They actually extend around the lateral domain in areas where you don't see much, if any, tight junction. And this is a magnification of this. And that led us to wonder whether the role of the tight junction in the phenotype, which then leads us to Natalie. And regrettably, my faux pas, I have to describe the movies. I can't show them to you. So what Natalie works with is live imaging in 10-gram, or I guess even sometimes smaller mice. And these animals then, their liver is placed on is open, the abdomen's open, they're anesthetized, they're placed on a screen, temperature is controlled, and they're imaged microscopically, live cell, and then images are combined and processed. And so here's an example of when the wild type animal was injected with, they're both injected with a rhodamine dextran, which stays in the blood space. And you're not going to see it, but carboxyfluorescein diacetate, which normally is traveled through the blood, taken up into the liver, deacetylated to form carboxyfluorescein, which is intensely fluorescent. And that's a substrate for an ABC transporter, ABCC2, which pumps it out into the biocanalicus. And what this movie would show, and I'll replace it for other purposes so others can see it, is that in the area between the cell, you would see this fine, almost herringbone-like distribution of the biocanalicus. Now, this is interesting despite the absence of the movie, because in the LKB1 knockout, you see evidence, here's the rhodamine dextran, and this is fluorescein that still remains within hepatocytes and very small amount ever gains access to the canalicus. Furthermore, it was observed that much of the color in the vascular space was not just red or green, but there actually were large areas of yellow, wondering how does this fluorescein get back out into the cell, out of the cell into the blood. The two transporters on the basolateral membrane that could do that are not overexpressed in this cell, so we thought that it may be related to what happens at the tight junction. So here are components of the tight junction which collectively function to link the actin cytoskeleton with a fully impermeable barrier, which permits the formation of the apical domain. Here shows the fragmentation. This is the bile acid transporter stain. A lot of it's intracellular, and it's very fragmented. And that's sort of matched by Claudin 1 shows a similar pattern. In other words, the biocanaliculae is grossly distorted, and the canaliculi and the tight junctions are likewise highly fragmented. Now, cingulin, which links the actin cytoskeleton with ZO1, is totally disrupted. Now, it's interesting that cingulin has an AMPK consensus site, and that may be the mechanism by which this is happening. Now, regrettably, once again, I can't show you this. Huh? This one works? How do I get it to work? Oh, there we go. Okay. All right, so here's the wild type. This is the knockout. What you see here is dextran, 3 kilodalton dextran. It's a little complicated. There's another substance in there, but this is the vascular space. And the black area is where the hepatocytes would be. And between the hepatocytes is where the biocanaliculae is, but you don't see anything. But here, you see the vascular space, and you see this. So what is this? This is 3 kilodalton dextran that has gone from the blood directly into the biliary system. This is the first demonstration, to my knowledge, ever at the cellular level in an intact organ of the existence of the paracellular pathway, a way in which material gets from the bloodstream, from the canaliculus back into the bloodstream. And this has been proposed to be an extremely important mechanism in various acquired diseases where the biocanaliculae can be disrupted. Okay, so here are the key points which I've tried to present. From the standpoint of structural polarity, each hepatocyte contains apical membrane domain which is to 1 or more, sometimes up to 3. Now that imposes a whole new dimension of understanding intracellular events when you have 3 apical domains within a single cell. These canaliculi are functionally sealed by tight junctions, and they form a very complex interconnected network. Their surface area, normally they're only about 10% of the surface area of the hepatocyte, but it's greatly increased by the presence of microvilli on the surface. And we don't understand the dynamic nature of those microvilli. Now, functional polarity is maintained by the polarized flow of molecules across the canalicular or the basolateral membranes. Things are coming into the liver, going out of the liver from the blood side, and selectively going out of the liver on the biliary side. When the bile components, particularly the bile acids, are retained, either due to some genetic disease or potentially an acquired disease, that leads to cellular damage, to mitochondrial damage, and ultimately to structural depolarization. these are the elements of the polarization system, which I won't take the time to describe, as we mentioned them before a couple of times. I would dwell on the fact that mitochondrial energy production is essential for polarization, and any disorder, particularly an acquired disorder, which profoundly alters energy metabolism, has its downstream consequences of depolarization. And that process is highly regulated by AMPK. So there are many more questions than we have answered, and the state of the art in the liver is far afield from where it is in the neuron, but we hope that the twain will meet. And that's the purpose of sessions like this and this course. So thank you very much. So we have time for a few questions if anyone wishes. Yeah. Yes. No. No. The liver, yes. Yeah. But we don't know as much. You see, we don't know as much about the liver from the morphological standpoint because, you know, biopsies are taken of these children at a time when there's no pathology, inflammatory pathology, so you can see what's going on, and they usually don't do the biopsy of the liver. In the knockout mouse that has been generated, there are both liver and gut alterations. The syntaxin 3 is interesting because that's strictly a liver problem. Okay?
I really enjoy reading your talk. I was just thinking, what are your thoughts about the fact that hepatocyte does not permit any virus to get in?
I'm sorry, Patricia.
That hepatocytes does not allow hepatitis virus to get into the cell. Do you think that it is related all about polarization? That none of the hepatitis viruses can get easily to the hepatocyte?
Well, that's not quite true, is it? I mean, hepatitis B enters hepatocytes. Hepatitis A. Well, you're the expert.
Yeah, no, but it's not easy because you have to transfect. It's not like HIV that you take the virus, it gets into. I don't know if it's because of complex, you know, the hepatocyte with polarization. I don't know, because must be one reason none of the virus grow primary isolate.
So Juan, you want to comment a little bit about how viruses get into cells?
No, I don't know that.
Yeah. I mean, most of the work with viral entry has been with things like VSV and others, and so it's it's indirect. But I mean, it's quite clear that occludin and clathrin 1 are docking sites for hepatitis C. Now, how that happens and how it machinerizes and what the consequences are, I don't know. But we have some data which lead to the interesting hypothesis, a working model, is that when cells are chronically infected, like with hepatitis B, and they're loaded with surface antigen, many of those cells are depolarized. And so far, depolarization is not a healthy state of affairs. They don't seem to repolarize. They presumably are eventually going to be replaced. Yeah. So thank you all very, very much.
Thank you.